So now we will go for the second step, that is the secondary structure prediction. Now the word changes from analysis to prediction. Okay. Why this change occurs? Because if I say primary structure analysis, primary structure means nothing but sequence of amino acid. Okay. So when you go to the UniProt server, we are getting the sequence only. So there is no meaning of predicting the sequence. Already we have a sequence. So we are analyzing the primary structure. That is why it is called primary structure analysis. Whereas when you go to secondary structure, so in this topic, we have to predict the secondary structure because we are having the protein sequence. Now we have to find out what are the type of secondary structure the protein should have. Okay, so what are the possible secondary structure for proteins are usually a helix or coils or turns, loops. So out of this particular sequence, which part of the sequence is going to form coils and which part of the protein is going to form a helix. So that is what we are going to identify with these tools. Okay, so we will use secondary structure prediction tools, and we will give example like Sopma you can use. So there are so many tools are available for this secondary structure prediction. Again, we will give different tools name or simply secondary structure prediction. You will have the list of tools might be available listed. Okay, so we can use any of these tools for predicting the secondary structure. So first I'm going to use this Sopma tool. It's a secondary structure prediction. You can see secondary structure prediction. So again, when you go to this particular secondary structure prediction tool, you can paste the sequence, and if you want, you can give the name of the protein. Say caspase 9 we have given. Okay, so you can give this name and then you can submit. See, now it is going to predict the secondary structure of the protein. So this servers again are present in this network protein sequence analysis. So again, this is like XPASI. So like XPASI, this is also another server which contains more number of tools for protein structure analysis or sequence analysis. Okay. So now once the server complete the prediction, you will get the result in this page. In the meantime, we will go for any other server for predicting the secondary structure. There is also another server known as Gor, G-O-R. So it is also used for secondary structure prediction. We will check that too also. Okay, you can see now there is also another tool. Gor is also available. There are so many tools are available for secondary structure prediction. So you can see now. So this is what the secondary structure prediction from the Sopma. So result for caspase 9. You can see here, this is what our protein sequence we provided it, and this line indicates that the prediction. So you can clearly see this. The first 20 amino acid might form the helix. Second 2 amino acid will form coils. Again we will get helix. So like this we can see this, the explanation for the secondary structure. Alpha helix might be 38% of the protein sequence will have around alpha helical structure. Similarly, the beta pleated sheet is around only, extended strand is only 11%, whereas random coil is around 49%. So this is what the probability of different types of secondary structure which are available in the submitted protein sequence. So you can see this clearly that the protein might have more type of coils. Next to that, we will have an helix, and next we have a beta pleated sheet. So like this, we will identify the possible secondary structure of the protein, and you can also see this where the secondary structure is going to present, also identified with this particular server. You can see more of the helical regions. You can see the blue color indicates helical regions located in the N-terminal region. So only the other part, say in the purple color, you can see this. Here also you can give a sheet. The purple color is indicated by the sheet. So the sheets, beta pleated sheets, are only present in this middle part of the protein for around 100 more than 100 amino acids. That is after 100 amino acids and before 350 amino acid, you will have this particular sheet structure. So this is turn is also present in many places, and then coil is also present in the many places. So this is what the prediction of secondary structure from the Sopma server. So you can save this file and you can utilize it for the research purposes. Okay, similarly there is also NPS server. So here also you can submit your protein sequence and you can give. So almost the results might be similar. If we have the prediction of Sopma or GAR, you might have similar type of results you will get. See, this is also a Sopma server. So almost you will have get the same type of output from the Sopma as well as the GAR4. So GAR4 is another type of prediction tool and Sopma is another type of prediction tool. So they might have used different algorithms and parameters for the predictions, but the output looks almost similar for both the servers. Okay, so in Sopma you can get four type of outputs: helix, sheet, turn, coil. But in NPS you will get only helix, sheet, and coil prediction. So whichever server is available, and we can use any of this for the prediction of the secondary structure. So now we predicted the secondary structure of the protein by using Sopma server as well as the GAR4 server. As I told you, both of them are present in the NPS. So even GAR is present in this NPS as well as the Sopma is also present in NPS. So this is what network protein sequence analysis server, both things are present. Okay.